siguiente de charlas de la doctora Eva Nogueles, English professor de California Berkeley y Howard Hughes Medical Institute. Adelante. Gracias.
Muchas gracias. No sé lo que significa que hoy haya menos gente que ayer. A lo mejor no lo hice tan bien. Para la tercera no va a haber nadie.
Van llegando.
Vale, no, es de broma. Quería empezar hoy hablando de la segunda razón por la que estoy aquí, llevando mi sombrero, como dicen en los anglosajones, otro sombrero, que es el de colaboradora con el Pew Charitable Trust. Esta es una organización de Estados Unidos muy conocida allí, con muchos años, que se dedica a mejorar, cuyo objetivo es improving public policy, informing the public and invigorating civil life. Y entre las cosas que hace es promocionar funding para jóvenes promesa en Estados Unidos en biomedicina y también a latinoamericanos. Entonces, quiero hablaros de este programa que se llama Pew Latin American Fellow Programs. Tiene un advisory board en el que yo soy el chair y entonces estoy sirviendo un poquito como de embajadora de este programa. Estos son los 10 seleccionados del año pasado. Los de este año van a ser anunciados en unas 2 semanas, así que prestad atención. Siempre tenemos muchas nominaciones, no son nominaciones, perdón, son aplicaciones de estudiantes mexicanos. Dejadme que os diga en qué consiste. Esto es para gente que está a punto de terminar, ha terminado o recién terminado su tesis doctoral. Tiene como idea tener un grupo de investigación independiente aquí en México y quiere pasar una temporada, unos años de postdoc en Estados Unidos en un laboratorio puntero, aprendiendo nuevas técnicas o con nuevos modelos o de alguna manera extendiendo su conocimiento y sobre todo también extendiendo el número de contactos que tiene con gente en Estados Unidos. El programa da a los que son seleccionados una cierta cantidad de dinero para su postdoc, que el resto lo pone el laboratorio que recibe, pero lo más importante es que luego, una vez que estos científicos vuelven a su país, se les dan $70,000 por ahora. Esperamos que esta cantidad pueda incrementar en el futuro para que inicien su laboratorio. Pero lo más importante es de verdad que establecen todas estas conexiones. Entonces, voy a tomar unos minutos para enseñaros un vídeo.
Because it provides 2 types of benefits to fellows. One is the actual training that you get here in the US, and also access to networking opportunities, not only with scientists in the US, but also with scientists in multiple countries in Latin America. And it also provides a very unique opportunity that's not available with any other type of funding to start in Latin America.
Once I dig into the details about the program, I get very excited, and it was like my dream, my professional dream. This experience provided me a great opportunity to start my career. I want to take money and invest in my own country.
7 out of 10 people that are a fellow return to their home countries. So opportunities do not stop when you return. And that's something really valuable about how Pew is able to build a community, not only fund particular talks.
The Pew community is actually quite large. We have hundreds of biomedical scholars, and we have about 250 Pew Latin American fellows that have participated in the program. That network of researchers in Latin America is very important because it allows us to connect and try to do multidisciplinary research. So having access to those biggest minds in the fields is just spectacular and provides unique opportunities for young scientists.
Science today is something that is not built by a single lab in a single place in the world. It's not only interdisciplinary, but it's global. And Pew is helping that.
Doing research in Latin American countries can be challenging because of the lack of resources that we normally face. So having the opportunity to be trained in these countries where we have access to cutting-edge technologies has provided me opportunity to train very sophisticated instruments, which I won't have otherwise the opportunity to do it in my country.
I'm really thankful for having the EU funding while I was postdoc and later on in Argentina. It enabled me to build the lab, and now it is helping me to build collaborations that will get funded and will be the kind of interdisciplinary science that you need when you build.
When I go back to my community in Colombia, I just want to share all of the different tools that I have learned from here to promote the growth of young scientists.
I want to give scientists in Latin America the tools for that. If I was able to do it, you can too.
Scientists from Latin American countries, South America, Central America, all of them are invited to participate in this program. And actually, that makes this program very spicy.
We are looking for talented scientists that would spend part of their career in the US learning new things that they can bring back to build science in Latin America, and you can be part of it. Apply to become in Latin America.
Okay, I'm glad that played. So, so I saw Diana in Argentina last fall when I was visiting. Yesterday I was with Juliana Paula. They're two Latin American fellows that now have their own labs at UNAM. So this happens, and please consider it. It could be something for you. Okay, so at any time during the day when I'm here, if you want to contact me about this, I'll be delighted to tell you more. But now back to the theme, the scientific theme. Today I'm going to tell you how I use Cryo-EM, the methodology that I introduced to you yesterday, to look at very large transcription initiation complexes. And that's just like a very, very quick summary. I told you that we take our samples, purified samples, we freeze them in vitreous eyes. We obtain images that are very noisy, but by using many of them and computationally aligning them and averaging and then putting together different views, we can obtain the structures. And this, remember, allowed us to overcome the requirement for crystallization, be able to study fully assembled complexes. And with the new detect and software technology, we can really get now to in the last few years to atomic resolution and see individual conformations and all the rest. Here, I want to emphasize something that was very, very important for the studies, all the studies that I'm going to be showing you today, and is that we need very little amount of sample. So we need a couple of microliters of samples that are just a few tenths of nanomolar concentration. So this is unheard of for any other structural biology technique. Okay, so this is the system that I'm going to be telling you about. This is the human transcription pre-initiation complex 2, pre-initiation complex that includes POL2. This is the supramolecular assembly that has to come together at the core promoter of every POL2 gene in our genomes when it needs to be expressed. And it includes a number of very large complexes. The one that I'm going to be telling you the most about today is TF2D, a complex that is well known to some members of this audience. It is composed of the TATA binding protein and about 13 TBP associating factors or TAFs that make up a complex of 1.3 megadaltons in size. It also includes the polymerase, which is itself half a megadalton in size with 12 subunits, and DF2H, which contains both ATPase activity and kinase activity that are important for opening the duplex DNA and phosphorylating the polymerase. And it's also close to half a megadalton in size. And then there are other factors. Just let me tell you that we purify TF2D polymerase and TF2H from huge growth of HeLa cells using immunopurification procedures. And these factors are very rare, especially TF2D, so we get very small amounts, but Cryo-EM make it possible to characterize them. The rest, the smaller ones, we can produce in bacteria in larger amounts. They're not limited. So TF2D, what functions does it have? So TF2D has the capability of recognizing all POL2 gene promoters. Whatever sequences the core promoter uses, is capable of recognizing it, and in the process of recognizing it, is going to be able to recruit the polymerase and the rest of the pre-initiation complex. It also refers to not just as a general transcription factor, but also as a transcriptional cofactor or co-activator, because it's able to regulate the transcriptional level through interaction with transcription factors and with the interactions with a number of post-translational modifications in chromatin. But it is very hard to characterize structurally. We knew this going in, but we didn't know to what extent it was going to be difficult. So I told you already that we can obtain them in very small amounts. The complex is not terribly stable once you get it out of the cell. For transcriptional activity, this doesn't matter. You keep it in glycerol or in sugar or something like that, and the complex is fine. But in cryo-EM, if you have sugar or glycerol in the background, the density of the protein is the same as the density of the background, and then it's invisible. It's like camouflage, okay? But the thing that we did not expect going in, because there was no structural information, is that it's extremely flexible. And, I haven't seen this kind of flexibility in many other things that exist at equilibrium. I've seen changes that are very dramatic, like hemagglutinin or something like that when it gets in the cell or stuff like that. But just to, again, this is the complex. These are class averages that are put one on top of the other, making like a little movie, where there are three main lobes that we very imaginatively call A, B, and C. The B and C core is fairly stable, but lobe A, which is about close to half a megadalton in size, moves by over 150 angstroms. Okay, so structural biologists talk of flexibility when there is a domain that wiggles or a loop that moves around. This was large, and it made an analysis very difficult, especially in the beginning. Look at the year way before the direct detectors and all these fancy software techniques. So Mike, when he was a graduate student in my lab, now has his own lab at the University of Michigan, analyzed these type of images, and he was able to show the position of lobe A with respect to the BC core, and to show that it's a kind of bimodal distribution, with what we call the canonical TF2D having lobe A bound to lobe C, and the rearranged being one in which lobe A was interacting with lobe B. But I hope you see these are distances. These are very broad peak, and there is really a continuum because the thing is really moving across. The thing that got us really very excited is that when Mike gave TF2D core promoter and TF2A, which is a small transcription factor that helps it, bind to the DNA, he saw that this distribution shifted. So now most of the complexes were in this rearranged state. And then when he was able to obtain three-dimensional structures of a mixture of TF2D, DNA, and 2A, he was able to get to a structure. And it was very hard at the time with the software that was available. And I hope you can see that is this rearranged state, the one that binds the DNA. So this is a state in which all the DNA is being held like this between the C and A lobes, and this is something that can happen in this state, but absolutely not in this one. And this told us, and this is a hypothesis that we still need to prove, that the regulation of TF2D interaction with transcription factors could come from then binding and shifting the equilibrium, biasing the equilibrium towards this state, because these will more readily bind to the DNA. Okay, so we managed to publish this very well, but the resolution was about 40 angstrom. Think about it. If you were studying a protein that was 40 kilodaltons, 40 angstroms will be a sphere. Okay. Here, we had a story to tell, even at this resolution, because the complex was very large and very flexible. But it was very frustrating, of course. So eventually, what is happening with the lights? I'm going to have a bit. So eventually, we did get a direct detector. Remember what I showed you about the microtubules and how resolution improved. And Robert, when he was a graduate student in the lab, now he's finishing his postdoc at Johns Hopkins, used direct detectors, but also used a trick. that Yuan He, a postdoc in the lab, have used to reconstitute a simple pre-initiation complexes using TBP, in which we could purify complexes bound to DNA when we had them in very small amounts, okay? So something like SAS exclusion column is not an option, okay? So he used magnetic streptavidin coated beads and core-promoted DNA that was biotinylated at one end. Then he could add these in tiny little volumes, and then he could add excess TF2B and 2A, incubate, let them bind, then wash excess of whatever was not bound, use and then cap the DNA and with a restriction factor, and use that cross-link, the release complex bound to the DNA, mildly with glutaraldehyde, and then put that in the EM group. And using the direct detector, this is what Mike had got. He got this, okay? So remember, this is now the state, he's purified the stably bound complex. The resolution improved, but not hugely, okay? Things to notice, of this yellow low bay, the only part that we could see was this, which is bound at one end of the DNA. The rest was flopping around. Here, the density is shown at 2 threshold, so that you can see there is a part of low bay that is really moving around. And the moving around of that part was making very difficult the alignment of the images. So we used a trick at the time, which was to use focus refinement where that part of the complex is ignored. And that allowed us to improve the resolution. And the improvement in resolution was enough that then we could take the crystal structure of TBP bound to 2A and to Tata box DNA and fit it at this end. And once we had done that, we knew the position of other core promoter elements that we have included in the DNA, and those are marked with these colors, magenta, orange, and dark blue. And just by moving along the length of the DNA and knowing the distance in base pairs, we could call it the DNA set. Here are the sequences corresponding to these motifs. And I hope you can see that those are the regions where there is blue protein binding to them. But these are still blobs. The resolution here is about 8 angstrom or so. There are still blobs. But what we did was to use this trick. The resolution is limited because there's flexibility here, especially there's a hinge here. But what Robert was able to do is to do that focus classification again with that lobe that is very stably bound to the DNA. And then, very resourcefully, he found structures of homologous of one of the domains, the hit domain of one of the TAFs, and saw that there was a dimer. He saw that a bioinformatician had said that TAF2 was a structural homolog of a class of aminopeptidases, but having lost their activity. And then there had just been a crystal structure of a domain of TAF1 bound to TAF7 that the researchers have identified as a DNA binding element because they saw a Will helix domain that they could prove bound to DNA. So we put all of these in what we call docking at the time, hybrid methodology, and we were able to identify everything that was bound to the DNA. This little piece was not visible is a little folded domain out of a loop in the crystal structure that was invisible in the crystal structure, but that is stably bound to what is called the INR, which is the site where the transcription start site is by the. So this was really very satisfying because now we have the tabs that form this lobe, how they bound to downstream core promoter DNA. But after all that effort, we just have lobe C bound to DNA. We had no idea what was in lobe A and lobe B. Okay, so here comes Avi, who was a graduate student at the time and now is finishing his postdoc at Northwestern University. And he collected a huge data set of just TF2D by itself, okay? And then he did classification to see what kind of conformations he could see. And this is what is shown. So this was TF2D without DNA. He just superimposed this part on the structure of it bound to DNA, okay? So that you see where the DNA would go and where TBP would go. And these colors depict all the positions of low bay he was able to see. This blob is the region that normally binds to DNA that without DNA is really moving around. So this, again, this is made up of pieces that are always flopping, especially when there's no DNA. The thing that I want you to see, I'm going to tell you in a second that this knob there is TBP. And I hope you can see that there is a certain trajectory that the TBP in the lobe is doing that is kind of directing it to where it will finally be when bound to the DNA. And this, by the way, is the orthogonal view. It really is doing that kind of action. But in any case, in doing this, he split the data. So instead, he went back and put all the data together and treated the different domains separately and refined them separately. And doing that, he was able to get about 4 angstrom resolution for all of these and about 9 angstrom resolution for low bay, except for this part, which we know very well from when it's bound to DNA. So at the end, he was able to assign all of the densities and generate a model of where all the tabs were. And what really helped was the fact that low B and low bay are very closely related. And once he was able to model low bay could be deduced even at 9 angstrom resolution. And this is why. Okay, so this low B is made up of 6 histone fold domains that are in 6 different tabs. Okay, so histone fold domains that pair up between tab 9 and tab 6, tab 4 and tab 12, and tab 8 and tab 10. Okay, so this is histone folded domain that forming something like a hexosome in a nucleosome of histones. Okay, but these are tabs, and then they bind to tab 5, which is a protein that has a WD40 domain. This domain is called beta propellers, and then an all alpha helical N-terminal domain. I'm going to be telling you about this a lot. Okay, so this is a module that is almost repeated on the other end. But in the other end, instead of tab 8, which is there in a single copy, and is what connects this lobe more stably to the rest, instead of that, it has tab 3. And this tab 3, pairing with tab 10, then forms a platform for the binding of a 4th pair of histone fold containing subunits, which are tab 11 and tab 13. And those are the ones that bind to TBP. So by replacing one histone here and then adding this low bay now is the one that engages with TBP. Now, TBP is completely inhibited, totally inhibited. So the cavity in the saddle that normally will bind the DNA is bound by a peptide at the end terminus of tab 1, and by another one on the side that normally will bind to TF2A. And so TBP, in the context of TF2D, is inhibited. He cannot do his business. This is interesting, I think.
Eva?
Yeah?
And terminal domain of TBP is not located in the structure.
Yeah, so
It
what...
is known that it blocks the binding.
Yeah, so well, to be completely honest, the resolution there is just barely enough that we can take the crystal structure that exists of TF2D with the 2 N-terminal regions of tab 1 and fit it in. Anything that is flexible or has been competed away by something else will not be seen. Okay, so what I wanted to tell you is that these histone folds really make something that resembles a nucleosome. Okay, so here I put low A and low B aligned so that you can see a histone, a true histone core from a nucleosome, align it with the same color so that you can see the similarities. Okay, so this and this is the same except for this addition, and of course here you have a normal histone core. And you know, it was known from before that some of this stuff has histone folds that they could pair up, and this is a complex that binds DNA. So it had been proposed that there will be a lobe in TF2D that resemble a histone core that will wrap the DNA. And there's nothing like it in the structure. None of these histone folds have come even close. There's one of the tufts, TAP4, that has an extension that kind of touches the DNA, but none of the histone folds. And why is this? Because although they have evolved from histones, which have this very stable core, they have used it for structural purposes. And one of the things that have happened is through mutations, they have lost the positive surfaces that interact with the backbone, the negatively charged phosphate backbone of the DNA. So no, these things do not interact with the DNA like the histone core does. They just have utilized that to make now functional units that they can use for different purposes. So I'm going to summarize some of these concepts. Okay, these two lobes are built of this histone fold-like core-like structure, but the two lobes do completely different things, completely different. Okay, so lobe B is making part of the BC core that serves as a ruler, so that it positions certain elements at the right distance, so that when the core promoter sequences are recognized, they are at the right places, and with the start site also in the right place for later recruitment. Is also the lobe that interacts with 2A, which is going to be very important for the engagement of TBP with the DNA. Lobe A has as a function to keep TBP inhibited until the rest of the complex has recognized that the core promoter DNA for sure. Okay, and at the end releases TBP, lets go of it, and this is now the platform in which the PIC will build. So I'm going to go into more detail, because then what Robert and Avi did together was to collect a truly huge data set of TF2D with DNA and with 2A, what Mike did, but now with the direct detectors and with the new software. Not quite as good as what we have today, because things are evolving very fast, but much better. So this is what they saw. They saw the complex that is with nothing, and that has the lobe moving around. And again, those are the two things, the canonical, one that is extending, so in different positions. Okay, and this is what it will be in a situation where it has not engaged with DNA, and the TBP is completely inhibited by all of these contacts. Then they saw this, which we call the scanning, and it is a complex in which this lobe has engaged with the downstream core promoter sequences, and now lobe A is biased towards sampling the upstream DNA, looking for a site to bind. And in this conformation, given the geometry of the complex, the element that is inhibiting the DNA could start being displaced by the DNA just by sheer local concentration of the DNA that has been placed right by it. Okay, so we imagine this being something where we have flexibility, and the lobe is coming on and off because TBP is trying to bind, but it's not quite being able to bind. And when it touches it, it's not bending the DNA. We have another that is very similar, is the rearrange. It's the same as this one, but 2A is already there, and is really facilitating that TBP is in the right place at the right time. Okay, so again, TF2A, by binding to TBP in this geometry, is now just by action of higher local concentration, be able to compete with those peptides that otherwise inhibit from binding. And then finally, we see the truly DNA-bound state in which TBP has been released from lobe A. So this is one in which now TBP is bending the DNA. Is a site that has it's a state that has a very slow on rate, but then really slow off rate. Okay, and this is the one where it is bound to the DNA, and in the process of binding to the DNA, it competes away, is sterically hinders the binding of these elements, and that's why the complex, the TPP, is released from the lobe because it detaches from TAF 11 and 12. And what happens is now this, once it's detached, it frees the surface to which TF2B binds. And when TF2B binds, it can now recruit the polymerase to the site. Okay, so this is what we are proposing based on our structure. And now I'm going to summarize all of that and more that we did on the assembly of the pre-initiation complex, the recruitment of polymerase, the recruitment of TF2H, the opening of the duplex DNA, and the initiation of transcription with an animation that we did with Janet Iwasa based on our structures, but where we interpolate between our states and we also extrapolate a little bit. So this is the DNA. These are the core promoter elements that are going to be recognized by TF2D. This is the so-called class 1 nucleosome. This is a nucleosome with very specific post-translational modification that is placed quite precisely with respect to these core promoter elements, and that has histone modifications that are recognized by reader domains in TF2D that I haven't told you because they are very flexibly attached and we don't see them. We just know that they're there. Okay? So TF2D is very flexible. All these elements are moving around and the movements are realistic. The timing is not because we have no rates. We know that these elements, they're very flexible. The histone tails are very flexible. It is relatively easy for them to engage. And this, they provide, again, a local concentration of TF2D for the elements that have to make very precise stereochemical contacts reading the DNA. We think that this is also probably important to retain the TF2D for several rounds of recruitment of polymerase, maybe, of restarting. But we don't know. All right. So now the complex can really start touching the DNA and recognize the core promoter elements. Once this happens, the lobe is biased towards being in this location, and TPP can start touching the DNA. Remember, it's still inhibited, but these contacts can start being regulated. So TF2A, by binding to lobe B, can displace that kind of contact, and now TPP has been released, that site has been bound, and the PIC complex can be assembled. We studied that PIC assembly at that point, from the point in which TPP has been released. So this is what I'm going to concentrate on now. And we studied those building complexes one at a time, and then working with the full complex to study different stages through the transcription initiation process. So that's the platform that POL2 bound to TF2F interacts and bind to. In the process, there are a number of things that reorganizes, conformational changes, stabilizations that happen. Then TF2E binds. By the way, this sequential progress of how things added had been determined biochemically by people like Bob Rader. They didn't know why this was the right order. Now we know why, because every element that comes interacts with the last one added. So it makes full sense. And this is now the platform to which the large TF2H binds. This is a complex that we have studied by itself, as well as bound to the core promoter DNA and the rest of the PIC. And one thing that we've been able to describe is the large conformational changes that happen, both releasing the kinase module, but also moving the XPB ATPase, which is a DNA translocase, into the right position, activating it, so that then it can start walking on the DNA. As it walks on the DNA following the helical path, it not only moves the DNA, but also turns it. But the DNA is held very tightly here. So what is going to happen? The DNA is going to unwind. Okay? So as the protein walks with this tight, there is tension that is released by unwinding, and a transcription bubble opens that we saw is stabilized by elements, the polymerase, TF2B and TF2F. Okay? And this continues for a while until a certain amount of nucleotides have been added in the messenger. So that's my story. I just want to tell you that in addition, the CAC domain, the kinase module that contains the CDK7 that phosphorylates, for example, the polymerase, a process that is required for the polymerase to actually leave the core promoter and move into the body of the gene. It's very flexible. We purify it by itself, and we obtain the structure of it by Cryo-EM. And this, at the time, it was about 100 kilodalton. No symmetry was among the lowest thing that could be seen. We saw it at enough resolution, 2.5 angstrom, that we could see the binding of a particular drug that was in clinical trials, and this is at the resolution where we can start seeing water, and we can start seeing the kind of interactions that make this drug specific for this particular kinase and not others. Okay, so just for completion. Okay, preguntas. Juan Pedro, qué bien.
The angle bending was observed by X-ray diffraction.
Yeah.
It's the same with the...
It is the same. It is absolutely the same. So once the TPP is released, in the context in which we studied it, bound to A, to B, and the DNA, is superimposed. Yeah.
The mediator complex?
Yeah. So we have not studied the mediator complex. So Yuan He, who was the postdoc in my lab that did the study of the PIC, collaborated with Roger Tijan while he was in the lab to get the structure of the pre-initiation complex with mediator. Patrick Kramer has also done it, and the Shu lab in Fudan University have also done it. So these have been extensions from that work that include mediator. Yeah. Mediator is particularly important to place the kinase domain that I told you is very flexible in a position where it's engaging with the RPB1 tail that it has to phosphorylate. So that was one major. So when we did this study, there were structures of mediator bound to polymerase, and what we could do was to generate, I didn't put it here, to generate a model, just putting the pieces together of how that would look. We will also generate a model of how TF2D would be in the context of the PIC. All of those were eventually proven experimentally.
Mediante esta técnica es posible estudiar cómo afectan polimorfismos genéticos localizados en el promotor del DNA.
Ya, sí, es posible. Lo que pasa es que los estudios son más bioquímicos que estructurales. Quiero decir, lo que vas a ver es, por ejemplo, no sé si nosotros podríamos ver que hay ver bioquímicamente que la afinidad es menor y luego hacer este tipo de análisis para ver cuál es el elemento que tiene dificultad, en qué punto del proceso que describimos el complejo no puede seguir adelante o tiene dificultad. Eso sería más describir como diferentes proporciones de los estados con respecto a este promotor, que es un promotor ideal. Podéis imaginaros que buscamos el mejor que nos diera la posibilidad de visualizarlo estructuralmente.
Solo para entender la película completa,
Sí.
tú muestras el complejo y una pinza agarra el ADN y el complejo y desenrolla.
Sí,
Y
sí,
ahí
sí.
abres la base que se va a copiar en RNA. Pero después eso se tiene que mover río abajo.
Claro, por supuesto.
Entonces la pinza suelta, o sea...
Sí, este es la siguiente...
El foro...
Sí. Pues tiene que haber un momento en que estos complejos desenganchen y la polimerasa siga adelante. Ese proceso es bastante... no es trivial, de hecho, bioquímicamente se producen muchos mRNA mensajeros cortos antes de que la polimerasa pueda hacer el más completo, y es por eso, es como un, no sé, es un...
ensayo y error.
Ensayo y error, y también parte tiene que ver con el hecho de que estos son en reconstitución. No sé cómo de abundante esto ocurrirá en el contexto del núcleo cuando las cosas son más complicadas y está también el plus one núcleo, son mis cosas así. Pero sí, más allá hay cosas que tienen que desaparecer, tienen que soltarse para que la polimerasa pueda seguir su camino ella sola. Sí.
Sí, usted muestra que al inicio del complejo para unirse al DNA necesita de una modificación específica del electrón, ¿verdad?
No, lo que he dicho,
Ok.
lo que he dicho es que en el contexto del núcleo, en un promotor real donde existe un class 1 nucleosome, eso va a contribuir al reclutamiento y a lo mejor tener lo mismo, una concentración local más alta, pero también, quiero decir, los contactos que se hacen con el ADN son muy específicos y necesitan una la orientación adecuada de un complejo que tiene 1.3 megadalton, mientras que los RIDA domains y los tails, que tienen muchísima más difusión, se van a encontrar mucho más rápidamente. Y una vez que tengas eso, es más fácil, tienes más oportunidad, incrementas tu on rate por tener estos uniones que son mucho más fáciles de establecer. Nosotros podemos hacer la mayoría de nosotros hemos hecho estos estudios con ADN nada más.
Exacto. Que no
Solo hay un
sabía
estudio, un estudio que ahora tenga nucleosomas, pero en muchos otros casos, si vienes mañana, te enseñaré cómo ahora todos los estudios que nosotros estamos haciendo son nucleosomas y que son con nucleosomas modificados, que son los adecuados para los RIDA domains del complejo que nosotros estamos estudiando, o que sirven de factores reguladores, activadores, inhibidores. Entonces, ahora la siguiente frontera es todo con nucleosomas. Todo con nucleosomas. ¿Había algo?
Doctora, nos ha mostrado cómo el curso de este trabajo han ido refinando muchísimo la estructura de este complejo de la transcripción. Y usted nos comentaba la experiencia de cómo sus estudiantes han hecho diferentes bases de datos para hacer que van analizando y se van implementando una parte utilizando el software que es el más actual en esa época. Entonces, me gustaría preguntar su experiencia de sus estudiantes en cómo, cuando cambia el software, qué tan replicables son esos análisis.
Oh, no, perfectamente. Lo que hacen estos softwares es te dan la habilidad de separar tus datos más y más en distintos estados en vez de agruparlos, o sea, hacer la clasificación de forma más burda, de manera que en realidad no tienes un solo estado y todavía hay flexibilidad y terminas teniendo muy poca resolución. Entonces, lo que estos softwares están permitiéndonos hacer es esa clasificación, hacerla cada vez más fina, de manera que el grupo que estemos promediando sea más homogéneo y nos dé más alta resolución. Entonces, terminamos con mejor definición de los estados y más resolución para cada estado. Pero la consistencia es la misma, a más o menos resolución, con más o menos estado.
Y en base a ese mismo detalle, así como ha usted refinado mucho los estudios estructurales, también ha aumentado mucho la capacidad que tienen las computadoras para predecir estructuras a partir de
Sí, ahora lo utilizamos todo el tiempo. Entonces, sí. A lo mejor tengo tiempo de llegar a la parte final. Aquí ya no. Pero sí, ahora esto, acuérdate lo que estaba diciendo cuando teníamos esa estructura que estaba como a 6 o 7 angstroms y utilizamos homology modeling, estructuras que se habían predicho, que no sé qué son. Pues ahora eso, para cada una de ellas, haces AlphaFold. Entonces, lo que nosotros tenemos es que el AlphaFold hay un determinado tamaño donde empieza a tener problemas. Ahora vamos a empezar a investigar qué tal se le da uniones ADN y uniones ARN, pero todavía, por un tiempo, tenemos posibilidad de que la parte experimental utilice el AlphaFold para llegar a las conclusiones lo antes posible, para que puedas hacer el modelado de ciertas partes lo antes posible. Pero la descripción, AlphaFold, yo no me habré todavía jubilado cuando AlphaFold pueda predecir, ¿tú te crees las conformaciones del TF2D? Eso no va a pasar. Entonces, es la dinámica, es la interacciones inestables y reguladas que no va a poder, que todavía no tiene, porque es que no hay suficiente datos de base that
Más-
para que pueda utilizar esa información para hacer las predicciones. Pero ya todo el mundo utiliza. Los biólogos estudiantes estamos todos utilizando el fútbol todo el rato, todo el tiempo. Sí.
Y luego habría que considerar también las modificaciones.
Sí, sí.
So,
Mañana os voy a hablar un montón de eso.
hoy.
Perdón, hoy más tarde.
Pero yo encantado también mañana.
No, mañana tengo que ver museos y cosas. A ver.
TF2A is dispensable for transcription.
¿Perdón?
TF2A is dispensable for transcription.
Um,
And your model seems to fix TDP.
really facilitate. And it is not that established that TF2A is completely dispensable for people would say TVP is dispensable for transcription, but as you know, there are different forms, and unless you knock them all down, it's not going to be the case. I definitely, in all the studies that have been done, TF2A has played a big role to be able to shift the complex to a bound state. Okay, so I've been telling you about TF2D here, which is this complex that is going to recognize core promoter sequences, mostly through these non-histone fold related TAFs, okay, and TVP, and that was built with two scaffolds that are shown here in two pale shades of yellow. There is a factor, a transcriptional cofactor, that also have one of these histone fold scaffolds, and that is called SAGA. And it has a number of enzymatic activities. So it's a histone acetyl transferase. It acetylates histone tails, and it's also a histone deubiquitinase. It removes monoubiquitinations. And these activities are required to set up the right chromatin state for transcription to take place. So it is a transcriptional co-activator. It itself doesn't bind DNA. In yeast, it's been shown to include TVP. In humans, this has not been seen yet by any means. It doesn't mean it's not there. It means that under the conditions in which we purify it, or it's not there. It binds, is located, it goes to certain sites through interaction with activators, with transcription factors that have DNA binding motifs that will bring them to a particular gene that is being activated and recruits them, okay? So I'm gonna tell you about that complex now. When we started working on yeast, and we got scooped by the labs of Patrick Kramer and Patrick Schulz. So we moved to the human complex, which is always much more difficult. And this is just a schematic of the complex that I'm going to show you, where yellow is always this histone fold-like domain that resembles low B. TVP is always in red. There are these domains that are the ones that carried out these enzymatic activities. And in gray is a big protein that in yeast is called TRAP1 and in humans is called TRAP. That is the one that has the binding sites for a number of transcription factors, including oncogenes like c-MYC. Okay? We wanted to study human because there were a number of things were telling us it was going to be different. One is the conservation sequence is actually not very good. In addition, there are numerous deletions and insertions, with one with respect to the other, and there are a number of TAFs that the complex in yeast shares with TF2D that in humans are genes that have been duplicated and made specific for SAGA. I'll show you more about that. Human has an additional splicing module. It's still not fully understood why it is there, but it is existing in metazoans, but not in yeast. And in yeast, SAGA is not essential. It is absolutely essential in humans, okay? Is embryonic lethal if you get rid of SAGA, okay? So it is very complicated. As I told you, this is now I'm already showing you the structure. It has all these elements. This is just a schematic. I'm not going to be referring to all of these subunits, only to a few that are key for the story that I want to tell you. But let me tell you that in this case, we could not rely on pre-existing biochemical reagents like antibodies and competing peptides. But this is the era of CRISPR, and we are in Berkeley. Anyway. So Megan, when she was a rotation student, she ended up then not joining my lab and joining Robert Tijan's lab, but she continued collaborating with us. Tagged one of the subunits that is unique to Saga, which otherwise is made out of pieces that come from everywhere. Okay, that it shares with many different components. It's like a Frankenstein. Okay, but it has a number of unique subunits, and one of them she tagged. She tagged it with both halo tag and flag tag. The halo is because she works in her labs are labs that do single molecule, single cell imaging. Very, very sophisticated tracking of how these things move inside the nucleus. And the tag and the flag is so that we could purify it. Okay. And then Dominic, when he was a postdoc in my lab, he's now working in a company, biotech company in Switzerland, obtained the structure of human Saga. And this is what it looks like, and this is the yeast. There are things that both in human and yeast we see in great details, and others that we see very poorly, and they have low resolution because they move a lot, okay? And if you look at it, they look quite similar, but it's because I'm tricking you. You will see at the end that it's not quite so. So there are elements that are really, really flexible. They were very flexible in yeast, but in our case, in humans, they are so flexible, these two enzymatic modules, that we barely see them. We see them in 2D classification class averages as smears. But we know approximately where they are, in which part of the complex, but I'm not going to be telling you about it. There is a part which is where the splicing module docks that we see at low resolution, but we were lucky enough that there were a crystal structure of this because it's a module that it shares with an splicing factor that had been obtained. And then this is the density for the hit repeat in TAP6L, which is like the hit repeat that in TF2D dimerizes and brings together low A and low B. In any case, the rest we see in very high resolution, and that means that we can trace from scratch and get an atomic model. So let's start with this core module. I always have this schematic here so that based on the color, you know what I'm talking about. So this is the histone module that has is a core that resembles the nucleosome and the histone core of the nucleosome. And here there are the different components. I want to tell you there are certain TAFs, TAF L, TAF B. These are TAFs that are duplications of the TAF 6 and 9 that I told you exist in TF2D. Okay, and that then have a specialized for Saga. In the case of yeast, they have not. They just use it. They partition, they produce double or whatever, and they partition. TAF12 is the same. There are some that are more distant homologs, but that are some have some similarity to some of the TAFs that I told you about. And then these are the histones that would be like the equivalent. Okay, so there is something that resembles very closely what we had in TF2D. Okay, like in TF2D, this interacts with TAF5, which is in human is TAF5 like, type of L, because again it's another gene duplication. And remember, this had a WD40 domain and an N-terminal or helical domain. Okay, and then we have this protein. I'm going to call it yellow protein, okay, for simplicity. That is also part of that core. It's going to become relevant in a second, okay? This is the hit repeat that is connected to the histone fold domain of TAP6. All right, okay, and this is just to remind you that this is not wrapping DNA, it's not binding DNA. It has a structure that resembles the histone, but it's not, and it has a number of deformations and it's not DNA binding. So what I want to tell you is now I'm putting this in the context of the splicing module, which interacts with this hit repeat. And I'm doing this because I'm going to start telling you about the difference between the human and the yeast complex. okay? So this is the human, and this is the yeast. In human, this is where the green N-terminal domain of TAF5L is present. In yeast, it has moved by 60 degrees because the yellow protein is very poorly conserved and it doesn't interact with it. And instead, it's in a location that in humans interact with the splicing factor. So remember, yeast doesn't have a splicing factor. This one does. And the location of the splicing factor is generated by taking away that N-terminal domain and tag it with respect to the yellow subunit. Just for completion, if you look at how that domain is in the context of TF2D, is moved with respect to here in the opposite direction by 90 degrees. And I'm mentioning this just because these are modules that exist, that are kind of conserved, but in different complexes are used in a completely different way. They use those flexibilities to position them and get them involved in completely different kind of interactions. Okay? So now, this was the module that I was telling you about. Now I made it all white. I'm just showing the yellow subunit because in addition to what I told you, it has this long extension that wrap around this gray subunit, which is the trap module, the module that interact with all the transcription factors like CMYC. It has this very long extension. It's wrapping around and it's grabbing. Well, more comparison. So this is human. Again, I'm taking that lobe and I'm just now coloring two subunits. That yellow one that goes down is a different view, and the cyan one, which is TAF12. And I'm now going to show you the yeast. So I hope you can see the yellow subunit in yeast ends there. That's all that is present. In human, it has this very long extension that wraps around. But yeast has a TAF12 that is longer. And with respect to that one, it has this long extension that also wraps around trap and is grabbing it, but in a completely different way, following a completely different path. Now, this module is very tightly bound to the trap. This one has like a little pocket in the middle, so it's much more flexible in yeast than it is in humans. But now I'm going to show you the real surprise, because I've been fooling you with these representations. I'm going to really show you how different these complexes are. So I'm going to concentrate on just the core. That core that has the histone folds and the TAF5, and that is very similar in body yeast. So we can superimpose them, okay? And if you superimpose them, they look very much alike. This is exactly what you would expect from subunits that are highly homologous, okay? Remember, the yellow is much longer, but the rest is very much. And now that I've superimposed that part, I want to show you the rest. And you will see that this common module is interacting with trap, the human, on the bottom, and with the yeast on the side. It's interacting with the same part on the trap subunit, but using different elements of this conserved core module, which I think is absolutely remarkable. So in the process of evolving, they conserve the elements, but they rearrange them, which I think is very curious. And the last thing that I want to show you, now I'm concentrating on the trap module that is engaged by the yellow subunit, okay? And because now this is human, so we can take all mutations that exist in human genomes that give rise to, in some cases, you know, for example, this one is cancer, but in many others there are anomalies, problems during neurological development, and we can map them. And they map to different sites in trap, which are the sites that interact, among others, with these transcriptional activators. We give them kind of a specificity for what it is that they're affecting. And we can map them, we see how much they're exposed and not. They could be changing the interaction, they could be changing the conformation. But I'm gonna just give you two examples, okay? That are, you know, single point mutations that give rise to neurological, neurodevelopmental defects, okay? And these two are located in that yellow protein as part of how it engages with the trap. And they're on regions that do not even exist in yeast. So I tried to convince the Nature editor that human needed to be published in Nature, but we didn't. It went into NSMB. In any case, so the work that I show you on TF2D was started by Patricia, actually, when she was a postdoc in my lab. She became a staff scientist. All the purifications of these complexes are done by Jay Fang, my lab manager. These were the 3 sequential biophysics graduate students that did the impossible, which was really describe TF2D at a time when the resources were minimal. And then Yuan worked on the PIC, and Basil, who now has his own lab at in London, was the one doing all the work on TF2H and the CAC module. And then the work that I showed you was done between Megan and Dominic, and I told you that Megan is just finished her postdoc with Xavier Darsak and Robert Tijan, my colleagues at UCL. And there's 2 options here. One is I finish, we have questions, and the other is I tell you about something else. But remember, we're going to have another story. So, questions.
Una pregunta general que no sé ni siquiera si tiene sentido, pero para yo entender. Dijiste que Saga era indispensable en humanos.
Sí.
Y no lo es en levadura. ¿Esta información estructural te puede dar una explicación para eso o la historia es más compleja?
I think la historia es más
todo lo que
compleja.
saben.
La historia es más compleja. Yo creo que tiene que ver con cómo de importantes son algunas de las modificaciones que pueden hacer la deglutinación, la acetilación de determinados histonas en el contexto del desarrollo humano y de yeast. Yo creo que cuando en yeast tú tienes que decidir si eres a haploid, a diploid, espora o no espora, y eso es todo lo que tienes que decidir. La complejidad de los networks de gene expression no es tan elevada. Entonces, este tipo de factores que están contribuyendo de manera tan importante, quiero decir, a single point mutation, que está diciendo, tiene problemas de desarrollo neurológico, pues al cerebro de yeast no le importa. Entonces, quiero decir, también lo he mencionado muy rápido porque todavía no lo sabemos. ¿Cuál es la función del splicing module? Es un misterio. Y eso no es, yo no creo, aparte, nosotros lo único que podemos decir es por qué existe en un caso y no en otro. También splicing no es tan importante, o sea, existe, pero no es tan importante en Baring yeast. Entonces, también puede haber una contribución, pero no, nosotros podemos explicar el efecto de ciertas mutaciones a nivel estructural, pero algo como por qué es esencial, yo creo que es algo más complicado que tiene, donde habría que hacer estudios, pues eso, de expresión genética a distintos estados para ver qué es lo que está fallando. Sí.
¿Han tenido la oportunidad de seguir estas mutaciones en la proteína a lo largo de la citogenia, en los metasuarios, y ves si hay alguna correlación con la evolución del sistema nervioso?
No, es muy buena idea. Y supongo que hay gente que se dedica a esto por a living y que probablemente lo está haciendo. Es muy buena idea, no se me ha ocurrido. Porque hay mucho que ha pasado entre la levadura y nosotros. Y nosotros no hemos seguido explotando ese tipo de información. Pero ese es el tipo de cosas, como no podemos hacer todo y hay otra gente que lo hace mejor, que lo ponemos ahí, ellos lo tienen y lo pueden analizar. Pero tienes mucha razón, desde el punto de vista de la evolución, sería muy interesante ver qué es lo que está pasando con estas adiciones tan largas de una proteína que tiene este tipo de fenotipos a nivel de evolución. Ya.
Nada más, una pregunta extraña, pero que en levadura la proteína amarilla tiene un pedazo que humanos trae.
Sí.
A nivel de DNA, bueno, veo que toda la parte de conexión es como un polo que no está, bueno, un polo que no tiene forma secundaria definida, parece.
No, excepto por un dominio que viste al final, que sí, ese está muy bien definido.
Entonces, a nivel de DNA no cabe la posibilidad de que haya habido, por ejemplo, una mutación en el codón de determinación primero, porque toda esa parte parece como aminoácidos.
Estoy haciendo una pregunta muy parecida a la anterior. Probablemente no lo hemos mirado. No sé si esto se ha añadido en un determinado momento en metazoans o se ha quitado en Barullis. Eso es algo que, sin hacer más análisis, no podemos saber. Sí.
Creo que TFL es la proteína que tiene el dominio WD40. Entonces la pregunta es, ¿se sabe si hay interacción entre los dominios WD40 para que haga dímeros y eso pudiera interferir con la interacción o modular la interacción de la proteína?
¿Que haga dímeros en qué contexto?
Entre ellos, porque tienen dominios WD40 y esos dominios hacen interacción.
Bueno, los W40 son dominios de interacción entre proteínas, pero no necesariamente de dimerización. Es de interacción con muchas otras cosas. Por ejemplo, en lo que os voy a contar dentro de un momento, se utiliza para reconocimiento de péptidos de histonas. O sea que
Pero en algunos casos sí se llega a ver interacción.
nosotros no hemos visto dimerización de ninguno de estos complejos. Y se habrían sido muy notables, porque esto son 1.5 millones de Dalton, veríamos algo de 3 millones. Pero
Y
no.
eso no modifica la interacción con la proteína amarilla, porque está como unida.
¿Quieres decir en el type 5? No hemos visto ese motivo está interaccionando por un lado con el histone core, con este histone-like core, pero no lo hemos visto interaccionando con otras cosas. Sí.
Sí, la proteína amarilla tiene como partes que son ordenadas, ¿no?
Sí.
Pero algunas que no. No sé si en el modelo...
Son todos, por cierto, ¿eh? O sea, todo lo que os enseño siempre son los elementos que están ordenados. La mayoría de todas estas cosas tienen elementos algunos cortos y otros larguísimos, mucho mayores de lo que vemos, que están desordenados. Algunos de estos se ha propuesto incluso que contribuyan al reclutamiento en phase-separated dominios en el núcleo y cosas así. Lo único que vemos es lo que está estabilizado y es consistente.
Okay.
Todo lo demás no lo menciono, pero sí, prácticamente en el 100% de los casos hay algunos residuos que no vemos.
Okay, porque es como, por supuesto, es una foto, ¿no? de lo que está pasando.
Pero eso siempre, en todos los biólogos estructurales, lo que os enseñamos es la parte que está estable. O sea, hay partes que a lo mejor son flexibles, pero que luego le unes algo y se estabilizan, y hay partes que son siempre flexibles y a lo mejor tienen una labor en este tipo de phase separation y cosas así. Tienden a ser las partes menos conservadas.
Ok, pero es el hecho de que haya justamente como estas regiones como más desplegadas, como más como, y otras que son como alfa hélices, como muy, pero en números muy específicos, es porque es la interacción con la proteína, con su pareja, que lo hace que se forman eso. Claro. Hay como dominios, que son de manera local, hay dominios que le organizan la proteína en estas zonas.
No hemos estudiado el proceso de biogénesis de estos complejos. No hemos estudiado el proceso en el que un componente se une al otro y si lo hace primero con un dominio y luego el otro. No hemos estudiado nada de eso. Piensa cómo purificamos y la cantidad de material que nosotros tenemos, que tenemos que estar concentrando la muestra en la rejilla del microscopio. Hemos purificado y lo que tenemos es lo que está establemente unido. Todos los procesos intermedios en el ensamblaje no lo tenemos. Y entonces habría, por ejemplo, yo creo que reconstituir el proceso de biogénesis se podría hacer con un pequeño elemento, con una pregunta muy específica. Si tengo el TRAP y le doy el SAP12, la proteína amarilla, y utilizo hydrogen deuterium exchange with time resolve, puedo ver que se une primero, que se protege primero, pero para eso hay que hacer más procesos de reconstitución sistemas más sencillos con la técnica adecuada.
Esta diferencia de que se una un extremo de manera lateral, en el caso de levadura y abajo,
¿qué implicaciones tiene? Sí.
Sospechan ustedes que
Sí, por ejemplo, una de las cosas que hemos visto es, claro, es siempre difícil cuando un resultado es negativo, no puede estar nunca seguro, pero el saga de levadura une TBP. Entonces puede contribuir a la unión al ADN de forma más directamente y eso haría que tuviese una orientación bastante definida con respecto al promotor. Entonces, nadie ha conseguido, no se ve la purificación. Cuando nosotros le añadimos TBP en este no lo vemos. Lo hemos hecho cuando también tenemos factores de transcripción en caso de que, por ejemplo, añadir CMYC, cambiar la conformación y ahora TBP se uniera. Nadie ha conseguido ver TBP. Entonces, la posición en la que TBP está, TBP se une a través de dos proteínas en BADI-Gs, una de las cuales no existe en el complejo humano y la que existe está en una orientación completamente distinta con respecto a TRAP, que es muy difícil imaginar que una TBP. Entonces, esa reorganización podría tener que ver con poder o no asociarse a TBP. Pero en general yo creo que lo que está haciendo es posicionar los elementos que son funcionales, los módulos enzimáticos, en distinta orientación con respecto a la unión al factor de transcripción y a la posición del promotor. Entonces, algo de ese sentido.
Como lo de complejos.
Sí, entonces tienes elementos que hacen distintas funciones y cambia su orientación. Entonces, como Saga, cuando se une a un promotor en el contexto de cromatina, poner esto en distintas orientaciones puede tener un efecto. A lo mejor es muy, quiero decir, es muy settled y es importante, son distintos promotores y cosas así.
Es que a lo mejor ahí estaría el secreto de por qué tienes este efecto degenerativo del sistema nervioso durante el desarrollo. Porque si pierdes esta capacidad de reconocimiento con estos factores como son somos o factores de transcripción importante para el desarrollo neural, pues vas a tener aquí esta...
Sí, todas las superficies de TRAP expuestas son muy similares, que son las que intervienen, porque la región de TRAP que interacciona es la misma, es que interacciona con distintas partes del módulo central. Entonces no lo sé, es posible, podemos especular, pero eso va más allá de... Yo creo que paramos, hacemos un biological break y nos vemos para Polycom, donde voy a hablaros mucho más de nucleosomas y de nucleosomas con distintas modificaciones, porque es de lo que se trata.